Okay, so this is our fourth and final lecture in this series of neurophysiology, basic neurophysiology. And today we're going to talk about the central nervous system and the support cells. So it's primarily the part of the nervous system that's not neurons. And then there's some systems that I think warrant mentioning so that they don't get missed during the basic neurophysiology, as you'll be talking about them again when you get into pharmacology as well as neurology. So a little bit of a hodgepodge, but the focus is the central nervous system and the support cells. So if you recall, the first lecture we talked about what actually makes up the central nervous system, and we've been talking since that time about the electrical cells, the neurons. But more than half of the brain is actually not neurons, but support cells. In addition, mentioned on the first lecture, one of the first slides, that the central nervous system could be defined as everything within the meninges or within the blood-brain barrier. So we'll talk about the blood-brain barrier finally. I'll mention the meninges. And then we'll talk a little bit about spinal fluid, which is not a cellular component, but an important component of the central nervous system. So our learning objectives for this lecture is to understand the support cells, to know what the function is of astrocytes, oligodendrocytes, Schwann cells, microglia. I don't have it listed here, but we'll mention ependymal cells as well. We're also going to understand what the meninges are, the three different meninges that encase the central nervous system. We'll learn about cerebrospinal fluid production, circumventricular organs, and the blood-brain barrier. So understanding the function of all of these components of the central nervous system is our goal. Okay, so to go back to one of our original slides, we have the central nervous system. We're talking about the brain and the spinal cord, and the peripheral nervous system, which is comprised of the afferent or sensory neurons and the efferent, which is motor and autonomic. And you will talk about the peripheral nervous system starting at the next lecture with Dr. Furr. So the meninges are three membranes. So the meninges are just membranes, and they wrap around the nervous system, central nervous system. And the three from the outermost to the innermost are the dura mater, the arachnoid matter, and the pia mater. And the dura mater is very tough, and once you start dissecting brains in anatomy, it is the one that is, you can't really rip it. It is very, very inelastic. And so that is the meninge that we use to define central nervous system versus peripheral nervous system, anything outside the dura mater being peripheral. The arachnoid is the middle layer, and the layer that is adhered to the surface of the brain and the spinal cord is the pia mater. Between the arachnoid and the pia mater, between the arachnoid and the pia mater is a space, and that space is called the subarachnoid space, and it is where the central, I hope I don't do that throughout the entire lecture. It is where the cerebrospinal fluid travels, is in the subarachnoid space below the arachnoid, between the arachnoid and the pia mater. Okay, so those are the meninges. Let's talk about the support cells. So again, the support cells include the astrocytes, oligodendrocytes, the Schwann cells we'll mention. These are peripheral support cells, the microglia, and the ependymal cells. So let's start with the astrocytes, and they have many, many jobs. So they're called astrocytes because of the way they look. This is a picture of the fluorescent marker used to identify the astrocytes, and you can see they look like stars, and thus the word astro, so cells that look like stars. And so one of their jobs, and this was for quite a while thought to be their only job, is to physically support the central nervous system and the neurons by making a scaffold. So the placement of the astrocytes dictates where the nerves will go. And as we mentioned, nerves are clustered by function in the brain and the spinal cord. We have tracts in the spinal cord that you'll learn more about. We have areas of the brain, nuclei, and clusters of cell bodies and tracts, and that are functionally defined, but their function is tied to their anatomy, I guess is how I want to say it. And so that is a function of the astrocytes. They're the scaffold, and they make territories in the brain, and they hold those neurons into their proper place. However, they do a lot more. They are also involved in neurotransmitter uptake and degradation, and we mentioned them very briefly when we talked about glutamate and GABA neurotransmission, that the neurotransmitter could be removed from the synapse by astrocytes. So we'll show that again in more detail here. They also are involved in mopping up potassium from the interstitial fluid. So the extracellular potassium is removed by astrocytes. They are able to provide some nutrient support to neurons. And in addition, they are able to help do some repair, and they're involved in neuronal plasticity, which is part of how we learn, how we sprout nerve terminals for memory. And they are part of the blood-brain barrier. And again, the blood-brain barrier, as we mentioned, is another way to define what is outside or inside of the central nervous system. So they're involved in the physical structure of the brain, and they do this by being closely associated with the synapses and by holding the neurons into a location so that anatomy and function is tied. They also provide metabolic support to neurons. So neurons are very terminally differentiated cells. Most of their structure is involved in having the appropriate components they need to move electrical signals, and so they have support in some of their metabolic functions from the adjoining astrocytes. So the astrocytes are capable of making glycogen, and that glycogen then can be used to help provide some glucose when it's broken down for the neurons. And so they provide the neurons with nutrients such as lactate for the neurons to burn. And so here is showing the blood, the blood-brain barrier, the astrocyte, and the neuron. And what you can see is that glucose coming from the blood can actually go into the astrocytes and can be made into glycogen, which can be then stored in the astrocyte, and when needed, that can be broken down into lactate, which can go into the neuron and be used as a fuel source. Certainly, glucose can go directly to the neuron as well, but having a store of glycogen can provide 5 or 10 more minutes worth of glucose or lactate energy. It provides 5 or 10 minutes worth of energy to the neuron, and so if you were to have an ischemic event, that glycogen can be critical in trying to keep the neuron able to do its job. The astrocyte functions to help oligodendrocytes make myelin. So although the oligodendrocytes are the makers of myelin, they require astrocytes to promote that function through a regulatory protein. Astrocytes can also help if there's an injury to nerve cells, and they can help by either putting a scar down, and a scar will hold that place so that an inappropriate neuron doesn't occupy that area, or there's not a space of extra fluid. You can have a scar, and that scar is made by astrocytes, and if there are cells that can regenerate, you may get some regeneration. So the astrocytes deal with injury, filling up areas that can't regenerate or helping with regeneration. The astrocyte end foot plate is part of the blood-brain barrier, and it circles around the blood endothelial cells, making a whole nother layer that is part of this barrier. And it helps then to keep things in the blood from entering the cerebral spinal fluid. So we don't want free exchange between the blood and the brain, because if you think about all of the things in the blood, some of those would be fine out in the periphery, but detrimental in the brain. I'll give you an example of that in a minute. The blood-brain barrier has several different components. The first is that the endothelial cells that make up the capillary wall have tight junctions. So out in the periphery, capillaries have fenestrations, or little holes that different solutes and smaller proteins may be able to wiggle their way through these little fenestrations. But we don't want that to happen at the brain, so the capillaries of the brain have tight junctions of their capillary wall or endothelium. The next piece that makes up the blood-brain barrier are the astrocyte foot processes, and then there are pumps within the blood-brain barrier wall that is able to pump things that are lipophilic and therefore can go through the membrane, things that like fat and go through the membrane that we don't want in the central nervous system, can be pumped back out by pumps called P glycoproteins. All right, so let's look at this now as a figure. In the first top figures, you can see on the left the non-brain or the systemic capillary, and the capillary is made up of an endothelial cell that makes a circle basically with the blood in the middle. And in the systemic capillaries, there's a little gap that allows things to escape, and those are the little fenestrations. In the brain, however, the capillary has tight junctions, so there is no fenestration. So things that are going to leave the blood to go into the brain will either need to be transported by a carrier, or if they're lipophilic, they can go through the membranes, but they'll have to go through the two endothelial membranes, and they'll have to go through the astrocyte foot processes to get out. And this is shown in the figure below, that the endothelial cells have a lot of different types of pumps in them, and so those pumps can then be regulated, and that's how things like glucose gets in, or electrolytes are moved. But this helps to keep things like proteins and amino acids out. And if you think to our neurotransmitters that we talked about in the last lectures, many of those neurotransmitters are made from either metabolic products or byproducts or amino acids. So if we let amino acids and metabolic byproducts in freely, we may end up with abnormal concentrations of certain types of neurotransmitters. So we definitely want to keep everything out of the brain in the spinal cord unless we carefully select it to be transported in. And here's just another figure with a little less noise on it that shows the brain capillary endothelium with the tight junction, and then circled with the astrocytes foot processes around it to give it yet another layer between the blood and the cerebral spinal fluid. And as I mentioned, there's many things that we want to regulate in the central nervous system. Amino acids as neurotransmitter, ions, because if we're going to have action potentials, we have to be quite careful that we have the concentration intracellularly and extracellularly that we need so that we can have our graded and action potentials. and resting potentials. And then hormones, etc., things that might be very important out in the periphery could wreak havoc inside the central nervous system. So we keep a pretty strict separation. The things that can get into the central nervous system fairly readily are those that can go through membranes. So these are lipophilic substances. And so to remove these things before they get free in the central nervous system, there is a pump. It's an efflux pump. So in other words, it takes something from the inside and pumps it out. And this is called P-glycoprotein pump. And so it's going to transport drugs and chemicals, hormones, so they may be endogenous or of our own body's making, or they may be exogenous, such as a drug we've taken. And they'll transport these out of the endothelial cell and back into the capillary. So if something gets into the endothelial cell before it goes all the way into the brain, it can be pumped back out. And the gene that codes for P-glycoprotein is called MDR, or multiple drug resistance gene. And this is going to be important in veterinary medicine because there are mutations in the MDR gene that are present in collies, Shelties, Aussies, and Sighthounds. Not all of them have it. Not all of them have 2 genes that are mutated. Some might have 1. But all of these can lead to susceptibilities to more of certain drugs or chemicals getting into the central nervous system because the pump is not efficient at pumping those items back. And so probably most of you will know right now a drug that collies and Shelties and Sighthounds are more susceptible to than other breeds of dogs, and that is our dewormer, ivermectin. But you'll learn all about this in parasitology, and you'll learn about it in pharmacology as well, in more detail. But there are other drugs as well that are P-glycoprotein substrates that we use in veterinary medicine. Quinidine and digoxin are heart drugs. Doxorubicin and vinblastine are anti-neoplastic drugs, so chemotherapeutics. Certain lipids are able to get in, and then steroids such as dexamethasone are able to get in and are substrates for the pump. So these things get into the endothelial cell, and then they're pumped back out before they ever get into the central. So this is the structure of ivermectin shown here. You can see it has a lot of rings, and anything with a lot of rings is going to be lipophilic and able to get through membranes. And so ivermectin is able to slide through membranes, but in a normal animal, it's pumped right back out by the P-glycoprotein pump. The way that ivermectin works is it is a high affinity agonist to glutamate-gated chloride channels. If you recall, the chloride channels in our mammals or vertebrates are GABA-gated chloride channels. But in invertebrates, they have glutamate-gated chloride channels. And so when the ivermectin binds, it acts as an agonist, opening up those chloride channels, causing hyperpolarization, leading to paralysis and death of the parasite. And so that's why we use ivermectin to kill parasites. But the trick is, or the problem is, that it is also a low affinity agonist at the GABA-mediated central nervous system receptors. And so if you get a high enough concentration in the central nervous system because the pump is not efficiently removing it, because there's a problem in P-glycoprotein, because there is a genetic defect in the MDR gene, then you can get enough ivermectin to actually cause central nervous signs in the vertebrate, the host animal, the dog, in the case of the collies that we're talking about. And what will you see with ivermectin overdose? Well, you would see overactivity of the inhibitory neurotransmitter in the brain. And what you see is anything from depression, drooling, vomiting, to weakness, to being unable to get up, to seizures. Eventually it can be as bad as causing coma or death. And so you will see depression of activity from the brain. I've seen this in horses, where a miniature horse was given an adult horse's dose of ivermectin, and it was sufficiently high to cause these clinical signs listed here. You may see it in dogs that have the mutation. So it doesn't have to happen in a mutated animal if the overdose is severe enough. Sometimes you'll hear of dogs getting it when they go out, when the client is deworming their horses with ivermectin, and it's apple flavored, and the dog is with them eating all the apple flavored ivermectin that hits the ground. If that dog happens to have an MDR mutation, you may see an adverse effect from that. Okay, so we've got this very good blood-brain barrier. It is due to our tight junctions. It's due to our astrocyte foot plates. It's due to our P-glycoprotein. There are places, however, and times that it would be good for the brain to know what's going on in the periphery. One way the brain knows is because of the nervous system bringing information in from afferent neurons. But we also have other things that the brain can monitor, such as the electrolytes, the fluid balance, hormone balance. These are all things that the brain should be able to respond to quickly. So within the brain, there are some areas where we have a non-intact blood-brain barrier. So we have a little area where the blood-brain barrier is leaky, and that allows these specific spots in the brain to monitor what's happening in the blood and therefore have a response. So the first three that are in red are all part of the endocrine system. So these are associated with hormone production. They're either glands or areas in the hypothalamus, which regulates the pituitary gland. And so these are areas where seeing what hormone changes are occurring is of importance. There are also areas, the next set are involved in electrolyte fluid balance. We'll talk a little bit about the chemoreceptor trigger zone, which is on the floor of the fourth ventricle. I'm not going to ask you to name the 7 circumventricular organs, but I do think it's important you know that there are some areas in the brain where the blood-brain barrier is not completely present. We'll talk about, again, the chemoreceptor trigger zone is important because we have drugs that work there in veterinary medicine. And we'll talk more about the hormone active areas when we get to endocrinology a little later in the semester in Phys 2. The second place that doesn't have a blood-brain barrier within the brain is the choroid plexus, and the choroid plexus is where cerebral spinal fluid is produced. So it's not a full blood-brain barrier there, and that is because cerebral spinal fluid is made by taking things out of the blood, filtering it, and producing the fluid that way. So you need to be able to access blood a little bit more readily, and so that is done at the choroid plexus. So again, the circumventricular organs are very vascular, and it's a place where the blood-brain barrier is not fully intact, and there are communication points between the blood and the brain and the CSF. They're involved in water, sodium balance, cardiovascular regulation, energy, and immune system. They're involved in fever, vomiting, and other things that might happen if we are exposed to a noxious stimuli. So this is an ancient figure, but I actually really like it, so I kept it as part of my slides. And it's showing the chemoreceptor trigger zone, and that is sitting on the floor of the fourth ventricle in the brain, and it has input then from the peripheral blood, so it can see drugs, toxins, uremia, which is toxins from your kidneys, bacteria and infections, and it also can get some signals of motion sickness from histamine, for example. If it's triggered, the chemoreceptor, so it's responding to things in the blood, trigger zone will trigger an electrical signal that goes to the vomiting center, so the emetic center in your brain stem, and it's the vomiting center that initiates the physical act of throwing up. So all the muscles that need to do their different things, and salivating and everything, retching, all of that is coordinated through the vomiting center. And the vomiting center decides to initiate vomiting based on information from the chemoreceptor trigger zone. You can also get direct neural input both from higher brain centers as well as from the periphery, so the heart and the stomach and other organs can tell the vomiting center that they're irritated and cause the animal to vomit. So you can have vomiting because of severe stress. That is because your higher cortex, your brain, your higher brain, has sent a neural signal to the vomiting center. You can vomit because your stomach's so stretched out, and that's going to be a direct neural input to the vomiting center. Or you can vomit because you just gave, the dog can vomit because you just gave the dog a big dose of chemotherapeutic for cancer, and that's going to trigger the chemoreceptor trigger zone to initiate a neuro output that goes to the vomiting center that initiates vomiting. So again, the chemoreceptor trigger zone is on the floor of the fourth ventricle in the brain, and this is where drugs, toxins, and other things work from the peripheral blood. And there are many receptors. Probably one of the most active are dopamine receptors, that when stimulated, they will initiate a signal that goes to the vomiting center. So here are a list of some of the receptors of the chemoreceptor trigger zone, and when these are activated through an agonist, activated by an agonist, they will induce vomiting. So I don't know how many of you have ever seen a dog come into the vet clinic and you need to induce vomiting, and the vet will give a dose of apomorphine. That is a dopamine agonist, a D2 dopamine agonist. And so when you give this drug, it will activate that dopamine receptor in the chemoreceptor trigger zone and cause the dog to vomit. It's very effective. Most of the time, we don't want our animals vomiting. It's a very common reason a dog or a cat might go to the vet is because they are vomiting. And so our anti-emetic drugs, those that stop vomiting, all work as antagonists at one of these receptors at the chemoreceptor trigger zone. So if you have motion sickness, you might put a scopolamine patch on. That works by antagonizing muscarinic receptors in the chemoreceptor trigger zone. If you have an animal that is receiving a chemotherapeutic protocol for cancer and is having vomiting as a drug adverse reaction, you may put that animal on Zofran or on Dancitron to try to keep them from being so nauseated and vomiting. Two other drugs you may be aware of is metoclopramide and Dramamine, and they both work through dopamine or histamine respectively. So knowing what receptors, neurotransmitter receptors, are in the chemoreceptor trigger zone, we can use that information to block those to help treat vomiting, depending on what the cause of the vomiting may be. Okay, so back to our astrocytes. We're not done with them. That was just all part of the blood-brain barrier role. Now, another role that we've already talked about is that astrocytes function to mop up the neurotransmitters that are released in the brain, specifically glutamate and GABA. The astrocyte, which is lying alongside of the synapse, has glutamate and GABA transporters. And so when you have a action potential come down your presynaptic neuron and glutamate is released, glutamate can be taken back up into the presynaptic neuron, but the major mechanism by which glutamate is recycled is by going into adjacent adjacent astrocytes. Within the astrocytes, the glutamate can then be broken down to glutamine, and that glutamine is transported back into the presynaptic neuron where glutamate can be synthesized and put into vesicles. So why all this trouble? Why are we have a support cell that helps remove the glutamate from the synapse? Well, the main reason is glutamate is highly toxic. If you have a lot of glutamate and you do not have astrocytes to help clear the glutamate, you will have toxicity to the pre and postsynaptic neuron. And so because glutamate is neurotoxic, we want to have a mechanism to get it out of the synapse. Glutamine, on the other hand, is just the amino acid that is not neurotoxic, so that is safe to shift the glutamine back and forth between the astrocytes. And astrocytes are not particularly sensitive to glutamate, whereas the neurons are. Another thing that the astrocyte's able to remove from the synapse is potassium, and potassium is also neurotoxic in high concentrations. And so astrocytes have a lot of potassium channels at the area of synapses, and they will soak up all that potassium and clear it from the extracellular space, and then it can be redistributed safely. And so you get a lot of potassium release during your neurotransmission, and the astrocyte can help remove that and redistribute it. So uptake of potassium and uptake of glutamate preserve the health of the neurons by preventing excitotoxicity and injury. You can, during times where you have insufficient oxygen, you may see excessive release of potassium or glutamate, and these will help keep that from causing harm. Another function of astrocytes is they are able to make growth factors, both brain-derived and glial-derived nerve growth factors. So they make growth factors that help neurons in both just maintenance function as well as repair. Also, if you're making new synapses, that process is known as plasticity, and that's how you learn. And when you learn, you make new synapses. When you have memory, you make new synapses. And that plasticity relies on growth factors produced by astrocytes. So they are pretty much the killer great support cells. They are the scaffolds. They make territories. They are involved in getting neurotransmitters out of the synapse and degrading it to a safe compound. They get potassium out of the synapse and interstitial fluid and keep it at a nice safe concentration. They hold 5 to 10 minutes worth of nutrition for the neurons. They are involved in repair and plasticity by making nerve growth factors, and they are part of the blood-brain barrier, which protects the brain and central nervous system from items that are being transported within the systemic circulation. Okay, so our next support cell is the oligodendrocyte, and its most important function is that they make myelin. And so in the central nervous system, myelin is made by oligodendrocytes, and the myelin then wraps around the axon of nerves and insulates them from any kind of injury. In addition to insulating or protecting the axon, it is also going to make neurotransmission faster. As you recall, when we talked about conduction, we said we could have saltatory conduction if the neurons were myelinated. And so saltatory conduction was where the nodes of Rambier are located between areas of myelin, and the signal jumps from node to node, and that makes it quite fast. So myelinated neurons are faster than unmyelinated neurons, and if you lose the myelin because of a disease process, the speed with which the signal can transduce along the axon is slower. Out in the periphery, myelin is made by a different cell called the Schwann cells. So Schwann cells make myelin for peripheral nerves, and oligodendrocytes makes myelin for the central nervous system. They're slightly different in how they wrap around nerves. In the periphery, one Schwann cell will myelinate one peripheral nerve. In the central nervous system, the oligodendrocytes are close enough to many neurons so that they'll wrap around several neurons. Not a big point. So again, conduction is faster in our myelinated nerves, where our voltage-gated sodium channels and our voltage-gated potassium channels are located only in the nodes. And when the signal reaches myelin, it just slides across the myelin, making it much faster. And I showed you this link to the YouTube video when we were talking about conduction in the earlier lecture. But you can look at it again if you wish. So myelin is not the only function of oligodendrocytes. Oligodendrocytes are also where iron is stored in the central nervous system. It is metabolized and stored in oligodendrocytes. In addition, pH is highly regulated in the central nervous system by oligodendrocytes, and that is because the buffer, carbonic anhydrase, which you learned about with Dr. Ranjan, I believe. The buffer is present in high concentrations in oligodendrocytes, and carbonic anhydrase takes carbon dioxide and water and converts it to hydrogen ions and bicarbs. So you're able to then buffer pH through this system. So oligodendrocytes, myelin, iron, and it's a buffering through its carbonic anhydrase activity. The next cell of the central nervous system is the microglia, and the important thing to remember about microglia is it's the immune cell of the brain. So we have tight junctions at the blood-brain barrier, which means that the immune cells from the periphery do not get into the central nervous system typically unless there is some sort of disease that allows them to cross over. So we have our own immune system in the central nervous system, and the microglia are the major cells involved in immune function. So immune function can mean a lot of things, and you'll have a whole class in immunity, immunology, immune system. But the immune cells clean up debris by chewing it up and eating it, phagocytosis. And so if you did have some cells die, microglia can come along and engulf that debris that's left behind from dead cells. If you have some sort of an infection or a toxin or some other event, these microglia can become activated and release a lot of immune compounds that function in helping protect the body from pathogens and toxic events. So these are called cytokines and free radicals. And they can also make antibodies. And so they can become antigen-presenting cells if needed. So all the functions of the immune system can be provided by microglia. And microglia in a non-immune stimulated brain is going to look quiet. And if there's an insult, they can become activated, and you can physically see the difference in what they look like. A is the inactivated, and B is the activated microglia. And so if an animal goes to necropsy and you see these activated microglia, that suggests that they had some sort of an inflammatory or infectious disease or a neurodegenerative disease, but they had something occur in their brain to activate the microglia. Now, it doesn't mean it just happened, because they can stay activated for quite a long time in some progressive neurodegenerative diseases. But if you had an animal and you were suspecting that they had some sort of a neurologic infectious disease, at necropsy you would be looking for these activated microglia. Okay, our last support cell we're going to talk about are the ependymal cells, and these are the little cuboidal cells that line all the ventricles. And one of their functions, besides being a barrier, is that these are the cells that produce, circulate, and absorb the cerebral spinal fluid. So the cerebral spinal fluid is produced by the choroid plexus, and the role of CSF fluid is to help cushion the brain and the spinal cord. So it's a bit of a shock absorber, and it also has the function similar to what the blood does out in the systemic circulation. It's involved in exchange of materials, so both bringing in nutrients, allowing oxygen to diffuse, and taking away waste materials of the cells of the central nervous system. The interstitial fluid, or the cerebral spinal fluid, helps exchange between the interstitial fluid and the blood. So let me say that again. The cerebral spinal fluid will take materials from the tissue and return it to the blood as far as waste, and take things from the blood that are important, like nutrients, and bring them to the central nervous system, tissue, brain, and basically brain and spinal cord. So the choroid plexus is the area where the cerebral spinal fluid is made, and the choroid plexus is in the ventricles. And so typically we have tight junctions of the choroid plexus and leaky capillaries, and that's so the choroid plexus epithelial, the ependymal cells are tight junctions, but we have leaky capillaries, and those leaky capillaries at the choroid plexus let things filter out of the plasma, and then these choroid epithelial cells can pick and choose what is going to go through the tight junctions into the CSF. Let's look at a picture, because I don't know if that made a lot of sense. So we have then a choroid plexus, which is in the ventricles. So this is showing the lateral and the third ventricles. And if we look at a higher resolution figure, you can see that there's capillaries, and those capillaries are leaky, and they're letting the electrolytes and nutrients go into this extracellular space. And then all of these need to go through the epithelial cells of the ependymal cells of the choroid plexus, and these epithelial cells have tight junctions, and so these nutrients and electrolytes must go through transporters through the cells to make it into the CSF. Okay, so the capillaries will leak, but it's not completely leaky into the cerebral spinal fluid, because it has to go through the epithelial cells. So CSF then will be an ultrafiltration of blood, and the CSF will fill up the ventricles, and it'll circulate through the ventricles in channels, and then it'll go from that fourth ventricle down the subarachnoid space, down the spinal cord, and all the way down and back up, and then it'll go around the brain. And when it's in the area of the subarachnoid space, there are villi, and these villi will remove CSF and transfer it into a sinus, and a sinus is just a location that blood pools, and so basically it is taking the CSF and dumping it back into the systemic circulation. So CSF fluid in an animal is exchanged 3 to 5 times a day. So we have circulation, we have production in the ventricles, we have circulation, and then we have removal of the CSF through these villi. Let me skip a few slides and I'll show you how it's absorbed. So the arachnoid villi project into venous sinuses, and then sinuses, veins, and lymphatics work to remove the CSF fluid. So it's absorbed from the subarachnoid space by pressure through one-way valves. And if you don't remove it, you're going to have excessive cerebrospinal fluid, and that will cause expansion of the vesicles, and that's known as hydrocephalus. So let's look at this picture. So you have your subarachnoid space, and you have these little villi coming off, and they go into a sinus, a venous sinus. So this is basically systemic blood circulation. It's a little area of pooling that can then be removed by capillaries. So when the pressure is high enough in the CSF, you'll get these little arachnoid villi will pinch off and transfer the fluid to the venous, to the systemic side, to the sinus. Okay, so when you get a little build of pressure, it's going to drive it mechanically to the systemic circulation where it's cleaned up. And so you're going to be making the cerebrospinal fluid constantly. If the pressure's low, you're not going to be removing it, but once you hit that equilibrium, then you're going to start absorbing it. And in a healthy animal, you're going to turn that over several times a day. So you make it in the ventricles, you remove it of these little villi from the subarachnoid space, and that allows you to turn over the cerebrospinal fluid, dump it into the bloodstream, get it all cleaned up out in the systemic circulation. And just a clinical application of some of what we were just talking about. Cerebrospinal fluid can be used diagnostically if we have an animal that we believe has a neurologic condition. And so we may need to sample that from our animals. And this slide is just showing two of the places we might go to get a sample of CSF. Particularly in our large animals, we may use the lumbosacral space, which is shown here looking on top of the back of the horse. Between the last lumbar vertebrae and the sacrum is a fairly large opening. It's fairly deep and not all that easy to access, but when you do access it, it allows us to take a sample to look for things like blood, protein, or cells, or bacteria. The other place we will commonly use to sample CSF fluid is the allanto-occipital space, and that is shown on the figure showing the head of the horse, where you're going to go between the nuchal crest and the in front of the atlas. And so in anatomy, you'll be able to see these spaces, and these spaces are in our small animals as well. Typically, we will use the AO or allanto-occipital space in our dogs and cats, depending on what we think is wrong with them. We may use our lumbosacral space more in our small ruminants than we might use an AO. And so these are just good anatomical locations for accessing CSF fluid.